Hello everyone, Vaibhav Shandilya here, Mbbs student from ESIC Government Medical College, Hyderabad. And today we're going to talk about the bloodstream infections and one of the most important organisms we're going to cover today, which is Rickettsia. And this is usually overlooked by students, but I personally got this as a 15-mark question in my examinations. So this is a very important organism. And there are a lot of classifications. There are different types. There are typhus subgroup and there is a spotted mountain fever subgroup. So there are different types of organisms in these, so we need to be clear about that. So we'll cover this discussion under the headings of introduction, antigenic structure, pathogenesis. Then we'll talk about the typhus group and the spotted fever group as mentioned. Then we'll cover the lab diagnosis and finally the treatment. So let's start with the introduction. So this Rickettsia is a gram-negative cocobacilli and it's an obligate intracellular organism. We'll reinforce this in the lab diagnosis, but gram-negative cocobacilli. Coming to the antigenic structure, we have two main antigens, the LPS antigen and the OMP antigen, outer membrane protein and lipopolysaccharide. So we have the LPS antigen and OMP antigen, and the LPS antigen holds a very significant role for the Rickettsia. How? The LPS antigen, it shares its structure with the strains of Proteus. Certain strains of Proteus it shares this antigenicity with, and this is the basis for the very important, and this is separately asked question, the Weil-Felix reaction. This is the basis for the Weil-Felix reaction. That is the diagnostic criteria for Rickettsia. So we'll talk about this Proteus strains, which are these, and the Weil-Felix reaction further in the lab diagnosis under the Weil-Felix test section. But for now, we need to know that the LPS antigen is shared by Proteus as well, which is the basis for Weil-Felix. Second, we have the OMP antigen, the outer membrane protein antigen, and this antigen is highly immunogenic. It's highly immunogenic, which is why it is used for vaccines. Okay, alright, so we have LPS antigen and OMP antigen. Okay, let's move forward. We have the pathogenesis next. Under pathogenesis, we'll talk about this in the form of a life cycle like transmission, spread, which the target site is, and so on. So let's start with transmission. So the transmission is by the arthropod vectors, and this is very important. Now, which arthropod vectors? We have, first of all, there are two ways of transmission. First by bite, and second by rubbing and scratching of the vector on the abraded skin. Okay, so the bite of the arthropod vector is by the tick or the mite. So it's kind of rhyming here, bite by tick or mite. And second, we have rubbing or scratching of vector on the abraded skin. This is by lice or flea. Okay, so we have rubbing or scratching by the lice or flea and the bite by tick or mite. You need to remember this. This is characteristic for the life cycle of each individual Rickettsia species. Now, after transmission, after we have entered the body, what next? So we'll spread. The bacteria will spread through the lymphatics. Through the lymphatics, it will multiply in the regional lymph nodes. After multiplying in the regional lymph nodes, it will spread via the bloodstream. So it's easy, transmission into the body, then spread through lymphatics, then multiply in the regional lymph nodes, and then spread via bloodstream. In the bloodstream, now the target site for the Rickettsia is basically the endothelial cell. Okay, it's the endothelial cell. So what this means is that on the Rickettsia, as mentioned earlier, we had the OMP antigen, right? So there are two types of OMP antigens majorly, the OMP A and OMP B. So Rickettsia adheres to the endothelial cell via this OMP A and OMP B. And after adherence to the endothelial cell, this is where the phagocytes then phagocytose these Rickettsia. Am I clear? So first we came in the body through the lymphatics in the bloodstream. Then the target site on the endothelial cell we bind via the OMP A and OMP B, and this is where phagocytosis occurs, and the Rickettsia has been phagocytosed inside the phagocyte macrophage. There, how will it survive? They have to survive. The bacteria has to survive to proliferate and cause the disease, right? So that is by a commonly used mechanism, inhibiting the phagolysosome function. That's it. Inhibiting the phagolysosome fusion. The phagolysosome fusion. If you inhibit that, the bacteria will not be killed. So they inhibit phagolysosome fusion and then they can multiply endlessly. They multiply via binary fission inside the phagocyte. Now, then after multiplying, after they have multiplied, they have to spread to other cells, right? To continue the infection. So this is a very important stage because this is different for both the typhus group and the spotted fever group. For the typhus group, what we see is the bacteria is multiplying by binary fission, right? After that, it will accumulate in the cell till it bursts. Characteristic. It will accumulate in the cell till it bursts, and after bursting, it will go and infect the next cell. Okay. And while in the spotted fever group, we'll see that the cell to cell spread or the migration is by actin polymerization. This is important. Now, actin polymerization is basically a technique by which, suppose this is the bacteria, right? This is the bacteria. It will polymerize its actin and form larger structures this side, and it will contract this side. So this part will go, and basically now it's like this, right? So it will polymerize then further and contract back from here, and then so basically it is now here. So you know how it is moving forward. This is how it will keep moving forward, by actin polymerization. This is the technique used. So for typhus group, by cell accumulate in the cell till it bursts and then infect the next cell, and in spotted fever group, movement or migration is by actin polymerization. So let's take a view of the pathogenesis till now. There is transmission first through the arthropod vectors, bite through tick or mite, and rubbing of the vector on the abraded skin by lice or flea. After that, we enter into the lymphatics, we multiply in the regional lymph nodes and gain access to the bloodstream. There, we attach to our target endothelial cell via OMP-A and OMP-B antigen, where phagocytosis occurs, and the bacteria survives in the phagocytosis via inhibiting the phagolysosome fusion. After inhibiting that, the bacteria can now multiply endlessly via binary fission, and it multiplies a lot till it has to spread to the next cell, which in the typhus group, it spreads via accumulation and then bursting, and in spotted fever group by actin polymerization. So this is the pathogenesis. You can take a look. Next, now we'll talk about the characteristic groups, which are typhus group and spotted fever group. Let's start by typhus group. In typhus group, we have two main Rickettsia types, one causing epidemic typhus and one causing endemic typhus. Okay, these are the two diseases caused by the typhus group of Rickettsia, epidemic typhus and endemic typhus. Let's talk about them one by one. Let's start with epidemic typhus. In epidemic typhus, now we need to remember these individually. Any one of these can come as a short question, or together we'll have to describe all of them in a 15-marker long question. So this is very important. So coming to epidemic typhus, this is caused by, the causative agents is Rickettsia prowazaki, very famous and important bacteria, caused by Rickettsia prowazaki, and the vector is lice. Now, as soon as I say that the vector is lice, we need to understand that the transmission must be through rubbing of or scratching of the skin by the contaminated louse feces. Why? Because this has been mentioned earlier here, right? That the transmission is by rubbing and scratching of the vector abraded skin by lice. So you have to relate these things. You have to remember that if the vector is lice, that means we have the transmission is by rubbing or scratching of the contaminated feces on the skin. Clear? So vector is lice, and then we have transmitted inside. And now what will be the clinical symptoms? So you'll note that the clinical symptoms throughout in all these different groups tend to be kind of similar. So keep one in mind and then keep modifying them as we go. So clinical features for epidemic typhus are fever, rash, myalgia, and CNS manifestations. Okay, so fever. In rash, we have a rash that characteristically starts on the upper trunk and it goes on the entire body except the face, hand and sole. Okay, except the face, hand and sole. And this is very important to remember because in Viva or in any exam, they can directly ask some rashes which spare the face, hand, sole, or which only infect the face, hand or sole. So here we need to know that the rash starts on the upper trunk and it spreads on the entire body except the face, hand and sole. Clear? Next we'll have myalgia, and then we'll have CNS manifestations like confusion and coma. Okay, so clinical we have fever, rash, myalgia, confusion, coma. Rash characteristically starting on the upper trunk, spreading on the entire body except face, hand and sole. Coming to the risk factors which predisposes us to the infection by Rickettsia, we have overcrowded communities, prisons or refugee camps. These are all closed spaces, and so Rickettsia can spread easily. So overcrowded communities, prisons and refugee camps. There is another point if you want to remember, the Brill-Zinsser disease. Brill-Zinsser disease is basically a recrudescent disease, which means there's reactivation of the acute epidemic typhus, due to decreased immunity or the latent Prowazaki. So the latent Prowazaki in the body, it reactivates and causes the disease epidemic typhus again, or we have decreased immunity, so now the Rickettsia can infect. So this is a recrudescent disease, Brill-Zinsser disease. If you want to remember, this is an extra point with epidemic typhus, where the latent Rickettsia will again come and cause the disease, epidemic typhus. So this is epidemic typhus. The point to remember, caused by Prowazaki, and the vector is lice, and transmission by rubbing on the skin or on the abraded skin, and transmission of the louse feces. Clear? Next, let's go to the endemic typhus. So the endemic typhus, the causative organism is Rickettsia typhi. Okay, the causative organism Rickettsia typhi, and the vector is rat flea. Now, when I say flea, it should come to mind the transmission would obviously be through rubbing or scratching of the flea feces on skin, right, or inhaling them directly, but mainly on rubbing or scratching of the flea feces on the skin. Clear? So Rickettsia typhi, rat flea vector, rubbing or scratching, and the reservoir for this are rodents. Reservoir where they grow are like rodents, like Rattus rattus. Clear? Rat flea, Rattus rattus. So reservoir rodents. And finally, the clinical manifestations are similar to epidemic typhus, as I mentioned, but they are milder. What are these? Will come again. Rash, mainly on the trunk, anorexia, myalgia, headache, and fever. See, there's a clear correlation between the two. This rash, mainly on the trunk, anorexia, myalgia, headache, fever. So keep modifying them. So this is endemic typhus, characteristically caused by Rickettsia typhi. The vector is rat flea, and the reservoir is Rattus rattus. Let's proceed. This concludes the typhus group. Now we'll go to the spotted fever group. The spotted fever group, the main disease which we encounter, the main condition we encounter is Rocky Mountain spotted fever, as the name suggests. The RMS fever, or the Rocky Mountain spotted fever. What do we have here? First of all, let's look at the cause. Similar, architecture for each of these species I've covered. So for Rocky Mountain spotted fever, the causative organism is Rickettsia rickettsii. Okay, Rickettsia rickettsii, and the transmission is by bite of derma-centered tick. Basically, a bite of tick. If you can't remember anything else, the bite of tick. Obviously, wherever there is bite, there is either tick or mite. So here, bite of a tick. Which one? In USA, it's commonly derma-centered tick, and in South America, it's ambyloma. Okay, the derma-centered tick is more important to remember. You find questions related to this in the USMLE examinations as well. So, derma-centered tick, bite, or ambyloma. This is the transmission. Causative organism, Rickettsia rickettsii. What is the reservoir? Again, reservoir similarly smaller rodents or dogs. Okay. Coming to the clinical manifestations, similar manifestations again: fever, headache, rash, myalgia, anorexia. Rash again on the extremities and trunk. Okay, it's all very similar. You can make these things up. So clinical: fever, headache, rash, myalgia, anorexia. Coming to complications. Okay, this is important here. There are complications related to Rocky Mountain spotted fever, and this is also one of the most important ones. So the complications are basically there can be endothelial vascular damage, increased permeability, which can cause edema, hemorrhage, DIC, or CNS involvement, say confusion or coma, whatever. So CNS involvement, DIC, hemorrhage, edema, increased permeability, or endothelial and vascular damage. These are the complications associated. So that's it. This was Rocky Mountain spotted fever. Causative organism, Rickettsia rickettsii. Transmission by bite of derma-centered tick or ambyloma tick. Reservoir, dog or small rodents. Clinical manifestations: fever, headache, rash, myalgia, anorexia. And complications, you can see endothelial vascular damage, increased permeability, edema, hemorrhage, DIC, CNS. Coming to the less significant ones, we have the Indian tick typhus and rickettsial pox. These are not that important, but if you know the causative organism and the vector, they can fetch you extra marks. So first, Indian tick typhus. So this is caused by Rickettsia conuri. Okay, what? Rickettsia conuri. And the vector is a tick bite. Okay, tick bite, Rickettsia conuri, and the clinical manifestations are S-jar formation, and the rest are similar to RMS fever. The ones we mentioned earlier. So basically, the thing you need to remember is that there can be an eschar formation, and it's because of a tick bite, and the causative organism is Rickettsia conuri. That's it. Now the last two both have an eschar. I'll state that later. Coming to rickettsial pox, the causative organism here is Rickettsia acari. Okay, you write these down multiple times and you'll remember all of them. It's not that hard. Just keep writing them down. Make mind maps if you want to. Okay, rickettsial pox is by Rickettsia acari. The vector is bite of mite. If there's bite, there'll be either tick or mite. We have mites here. So, caused by Rickettsia acari, the only mite out of all is here in rickettsial pox, the bite of infected mite, and the reservoir tends to be a mice. So there's a lot of rhyming going on if you want to form n in mnemonics, mice. And reservoir is mice, and the clinical manifestations are eschar, regional lymphadenopathy, and vesicular rash. What? Eschar, regional lymphadenopathy, and vesicular rash. Okay, these three you need to remember, and you will remember them eventually, because this is exactly the same clinical manifestations for a similar, the one I'll cover in the next video, scrub typhus. Scrub typhus has exactly a similar triad, which is vesicular rash, eschar, and regional lymphadenopathy. So you'll remember this. So this was basically all the organisms and the disease they cause. So in typhus group, you covered epidemic typhus caused by Rickettsia prowazaki, endemic typhus caused by Rickettsia typhi. Then in spotted fever group, we had Rocky Mountain spotted fever caused by Rickettsia rickettsii, Indian tick typhus by Rickettsia conuri, and rickettsial pox by Rickettsia acari. That's it. Keep revising them and you'll remember them for sure. Let's come to the lab diagnosis now. Coming to the lab diagnosis. Okay, so first, if you want to remember, we have Neal-Moser reaction. Okay, the Neal-Moser reaction is an animal pathogenicity test done on the guinea pigs to speciate the various rickettsial diseases. To speciate these various rickettsial diseases, we have the Neal-Moser reaction, which is an animal pathogenicity test done on the guinea pigs. Okay, coming to the most important thing to be covered in Rickettsia, the Weil-Felix test. This can be asked individually, they won't even mention Rickettsia, you just have to know. So this is a non-specific test. Now we'll talk about everything. So the Weil-Felix test, the principle is that the LPS antigen present on the Rickettsia is shared by Proteus. Not all Proteus strains. Characteristic Proteus strains which are OX19, OX2, OXK, we have to remember this under any condition. Which strains? OX19, OX2 and OXK. These three strains share the LPS antigen with Rickettsia. Okay, this is the principle. What will happen? Procedure. This is a tube agglutination test. Characteristic. Tube agglutination test. You need to remember the type. In general microbiology, they'll ask example of agglutination tests. You can add Weil-Felix reaction. It will come there as well. So tube agglutination test. What do we have here? Diluted patient serum is taken, plus we add the three strains of Proteus, or either whatever, the three strains of Proteus, sorry. So diluted patient serum, strains of Proteus, OX19, OX2, OXK, and what will we observe? How will we speciate the different Rickettsia types? If it's epidemic or endemic typhus, if it's the typhus group, we'll see agglutination mainly with OX19 only. Okay? Mainly agglutination will happen with OX19 if it's epidemic or endemic typhus. Coming to Rocky Mountain spotted fever, there will be an antibody to OX19 and OX2 elevated. Antibody to OX19 and OX2 will be elevated. So here it was only OX19 agglutination. Here antibody to OX19 and OX2 will be elevated in Rocky Mountain spotted fever. In scrub typhus, which I'll cover in the next video, it's basically a type caused by not Rickettsia but Orantia susugamoshi. Don't worry, we'll cover it in the next video. But if there is antibody to OXK elevated, then we'll see presence of scrub typhus. That indicates presence of scrub typhus. And finally, a negative test. We don't see any characteristic change. That means it's rickettsial pox. So, remembering this will really help you, really. So, epidemic and endemic typhus, we have agglutination mainly with OX19. Rocky Mountain spotted fever, OX19 and OX2 antibodies elevated. Scrub typhus, antibody to OXK elevated. A negative test for rickettsial pox. Clear? Now, we can get false positive results if there's an underlying Proteus infection, because then those strains will be there. No matter if you have Rickettsia or not, there will be a false positive. So, underlying Proteus infection. Otherwise, a false negative can be by a prozone phenomena where there's an excess antibody titer in the patient's sera. Okay, so that's prozone phenomena, which can give a false negative. Okay, so that's it for the wheel Felix test. Go through this again and you'll remember it. Next, in the serology, we have specific antibody detection tests. These were non-specific. In specific antibody detection tests, we have indirect immunofluorescence assay as the gold standard. Indirect immunofluorescence assay and ELISA. Isolation of the bacteria can be done. It's not cultivable. It's not cultivable. So you have to grow it on egg, guinea pigs, or cell lines like Vero or Hella cell lines. It's not cultivable. I believe this is an exceptional to the Koch postulate, which you'll learn in general microbiology as well. So you need to keep a note of that. It's not cultivable, but it can grow in egg, guinea pig, or cell lines like Vero or Hella. Clear? So that's it for the lab diagnosis. That's as important as it is. Treatment comes doxycycline. You can give for a treatment. Okay, doxycycline. And so yeah, this was Rickettsia. We covered it. This was a long video because this is an important one. This comes for a 15 markers. It's unavoidable. You will encounter this once in your second year MBBS. So let's just quickly recap. These are gram-negative cocobacilli. Antigens structure, what do we have? We have OMP antigen, LPS antigen. LPS antigen basis for wheel Felix. OMP antigen, it helps in the pathogenesis. We'll talk about it. And for vaccine preparation as well. Under pathogenesis, transmission by arthropod vectors, bite, tick or mite, rubbing or scratching, lice or flea. Next, spread, lymphatics, regional lymph nodes, and then bloodstream. Under the bloodstream, we attach to the endothelial cell via the OMP and OMP antigen phagocytose. There, survival by phagolysosome fusion inhibition. Then multiply by binary fission. After that, cell-to-cell spread for typhus group by accumulation and then bursting, and spotted fever group by actin polymerization. Coming to the typhus group, we have epidemic typhus caused by Rickettsia prowazaki, vector is lice. Here, endemic typhus caused by Rickettsia typhi, vector is rat flea. Rocky Mountain spotted fever, causative organism Rickettsia rickettsii, transmission by dermacentre tick. Indian tick typhus, Rickettsia conuri, tick bite. Rickettsia pox, Rickettsia acari, and infected mite bite. Clear? And then we had for lab diagnosis, wheel Felix test, Neal-Moser reaction. We had specific antibody detection tests and isolation. It's not cultivated. So this was the video on Rickettsia. I hope you liked the video. We have started Bloodstream. I plan on covering a lot of Bloodstream organisms and then make a playlist so it can help students, locate the videos better. So see you guys in the next video. Thank you.